Thank you, Steve, and thanks all of you for sticking around, and Agilent for allowing me to share our research with you this evening. So, if you take anything away from my talk tonight, let it be that you can use metabolomics to discover meaningful changes that you can then take forward to develop assays that answer your relevant questions. So a bit about Stemina to begin with. We were founded in 2006, and our expertise is in the areas of metabolomics, stem cells, and identification of small molecules associated with toxicity and disease. We use that expertise to develop diagnostics and predictive toxicology screens. And I'll be focusing on DevTox tonight, which is our teratogenicity assay. Also, currently in development, we have a diagnostic blood test for autism, as well as a cardiotoxicity screen that we're working on. Of note, the predictive toxicology screens are for compound prioritization, so basically for decision making. This slide shows the assay development life cycle at Stemina, and I'm trying to simplify a very complex process here. We begin with metabolomics and a distinct metabolic signature. Again, we're focusing on small molecules. We then create a model, and we further develop it using a training set of compounds or samples, then evaluate that in a blinded test set. Then we take that data and we further mine and refine our signature with the idea that we want to balance biomarker predictivity and detection. And we would like to refine the signature such that we can make it less complex. And at that point, we can develop an assay and then evaluate that assay, increase robustness and throughput, show reproducibility, and then take it to our application, which in our case is typically the market, commercialize the assay. We have a manuscript that we've submitted that goes over the DevTox assay that, if you're interested, you should watch for. So you might be asking, why develop an assay around developmental toxicity? Can't we do that already? So the scope of the problem is that there's 6% of births nationally in which birth defects are present. 5 to 10% of those are attributed to teratogens. And the rodent models that are used as part of the screen for that, and the regulatory hurdle actually, are only approximately 60% accurate and are many decades old. Here you can see this is our classic case of teratogenicity, the children of thalidomide from the 1960s. A very striking and dramatic example of the impact of a teratogen. In these decades, science has progressed, and our models can too. And in fact, in safety sciences, there's currently a paradigm shift wherein the vision is to use human in vitro assays to examine perturbation and toxicity pathways rather than relying strictly on animal models. So the DevTox assays, there are 2 of them, and I will tell you about how we moved from DevTox Discovery, our initial assay, which was metabolomics-based, to a biomarker-based assay, which is QuickPredict, which is very focused on the most predictive biomarkers that we found through our work with Discovery. The assays both use human pluripotent stem cells and enable compound prioritization through prediction of teratogenic potential. Quickly, the workflow, we began with cell culture, and it's in a 96-well plate format. We then expose the cells to compounds over 72 hours. They're dosed daily. An important point here is that we collect the media. We don't look at intracellular metabolism, but rather the footprint, the metabolic footprint the cells leave behind in the media. And another important point here is that we analyze viability. We don't want to be measuring the metabolomics or the signature associated with cell death, but rather a perturbation from the teratogens. That's a key part of this assay. Sample prep, we focus on small molecules, not the large, so we filter the samples to remove the large molecules. And then we use our LCMS-based platform to analyze the spent media, and our platform is based on Agilent QTOFs and TOFs and Hillet chromatography. The data analysis is where, well, part of where the two assays diverge. In the discovery assay, again, we're looking at many, many biomarkers, and we use that information to refine our assay down to just a few in Quick Predict, and you'll see why in a moment. So moving from omics to specific biomarkers, started with the discovery assay, our clients got a prediction of teratogenicity potential, cell viability, 3 exposure levels, and the assay took about 6 weeks. And it's a good assay. They also got information about metabolic perturbations, putative annotations, and pathway mapping. But we knew we could make it faster, and our clients actually wanted the assay to be faster to drive decisions faster. So the criteria was we were trying to still provide that prediction, which is crucial, provide cell viability over 9 exposure levels. More exposure levels helps you to put the prediction into better context in less than 2 weeks and at a lower cost. So our tactical plan here was to discover targeted biomarkers that we could then develop into a faster assay, change the dosing paradigm, and simplify the model that we use and with automated output as well. So finding that metabolic, predictive metabolic signature, this is the discovery method, and this is a total ion chromatogram. And you can see it's a long run. The scan range is from 70 to 1600 AMU, so over many masses. It's 30 minutes per injection, 2 injections per sample. We're running positive and negative electrospray, so this takes a lot of time. The first thing we have to do is determine what in that mess is most important. And to do that, we used our compound training set, so our DevTox training set, which consists of 23 well-characterized compounds balanced between teratogens and non-teratogens. And we did 9 independent replications of that training set, and that's a lot of data. So to give you some perspective, it's almost 2,000 samples, almost 4,000 injections. So we started, again, with our computation-based model, which looks at those many features of metabolites. We needed to determine what's most reproducible, and to do that, we developed an algorithm to evaluate the predictivity of all those individual features, and then looked at how predictive those features were in combinations. And at the end, we identified the most easy to measure and highly predictive combinations. This was a lot of work, but well worth it. So what we wound up with was ornithine, which is a secreted compound that the cells excrete when they're happy and healthy, and cysteine, which is a component of the media that the cells utilize when they're happy and healthy. And together, these 2 components were able to represent the signature while retaining the predictivity of the more complex computational model. This is part of our process, and I'm trying to put a lot of work into one slide here. So let's tell you what all these points mean. The red dots are teratogens in our training set. The black dots are non-teratogens, and our green line here is actually our fold change indicative of teratogenicity. And what that line represents is the measurement of ornithine and cysteine in a ratio. Using these 2 metabolites in a ratio allowed us to be 91% accurate when it comes to predicting our teratogens versus non-teratogens accurately. The sensitivity that we get can be attributed to the cysteine. It allowed us to be 83% sensitive, and ornithine allowed us a great degree of specificity. So you can see that most of our red dots lie on the side of teratogenicity. They're predicted correctly, as are our black dots. So what we wound up with through all of this work and effort was Quick Predict, which was a targeted biomarker assay. So from hundreds of analytes down to just 2, we were able to optimize our LCMS methods for ornithine and cysteine, narrow it down to 1 mode, electrostory positive, and incorporate stable labels, stable labeled internal standards, and that's huge. From going from a non-targeted LCMS run to being able to target and more accurately measure 2 metabolites was just incredible for us. With that, we were able to get faster. We shortened our runtime from 30 minutes to 6.5. And in the end, that allowed us an 8-fold increase in LCMS throughput. Remember, we're only running 1 mode now. It also contributed to the assay cycle time reduction from 6 weeks down to 2. And then once the assay was developed, we were able to show that it was highly reproducible by running a verification set of compounds. 17 batches over 120 days. Our internal standards and our analytes were well within the specs that we had set, and our predictions of teratogenicity were reproducible. So this slide shows how the output of the assay, essentially how the assay works. So the purple line is our measurement, the ornithine and cysteine plotted and fit to a line. The brownish sort of line here is our teratogen index. So we know that if our response falls below that line, a compound has teratogenic potential. And I want to show you a couple examples. Again, here's our friend thalidomide, a classic teratogen. And another thing that's important on this slide is viability. I mentioned that before. And here you can see the sensitivity of our assay relative to just cell viability alone. The cells are not dying. They're happy and healthy. They're not really changing much. But the metabolic perturbation indicates that this compound does indeed have teratogenic potential. And then I want to show you a non-responder as well, saccharin. Again, the viability does not change, nor does the ratio, as would be expected. So to verify our Quick Predict assay, this is just a slice of our verification set. We're publishing the full set. And I should also mention, we've looked at the assay in 60 compounds, I think over 60 at this point. And so we have a lot of data on many different types of compounds. But here I'm showing you a slice of what we tested, and some non-teratogens and some teratogens. The teratogen index prediction, which is Quick Predict, and the viability prediction. And you can see when you go down the teratogens how much more predictive the teratogen index actually is than cell viability alone. And some metrics are over here. The teratogen index is 97% accurate versus cell viability at 72%, and it's a lot more sensitive as well. And that is really a strength of it. So, as I mentioned, this assay is used for compound prioritization and is often used before a compound would be tested in the preclinical models that are accepted for regulatory purposes currently, rodent and rabbit. So we needed to know how concordant we were with these assays. If you get a hit in our assay, what does it mean in terms of whether you're going to get a rodent hit or rabbit hit? And so this second column is the number of compounds we were able to compare based on literature values. The third column is DevTox concordance, and you can see we're highly concordant with the animal models. This is, fourth column is the accuracy of the models as compared to DevTox at the end here. So we're more accurate, which would be expected. We're human cells versus an animal for this slice of compounds. And so this helps people to understand how predictive we are of the current regulatory hurdle. Also, we have on the slide the other in vitro assays and how we stack up there. So in summary, in our DevTox portfolio, we were able to go from a metabolomics-based assay to a targeted biomarker assay. We retain our predictivity very nicely. We're able to turn it around faster for a lot less money over many more doses to allow people to put the prediction into context better. And if you're interested in reading more about our research, here are two of our past publications. As I mentioned, we'll have another coming out soon. And I'd like to acknowledge the great team at Stemina and also Agilent for the technology they've provided us over the years, as well as technical assistance, and the NSF for their funding to allow us to develop and bring these products to market. And if you have any questions, I'm happy to entertain them. And I thank you very much for sticking around and for your attention.